Started with the name of Almighty Allah, the most beneficent, most merciful. So as you know, currently we have been dealing with pharmaceutical biotechnology, and under that I did talk to you. Introduction to biotechnology. In it we have to discuss genetics, genomics, proteomics, biomolecular target identification, pharmacogenomics, gene therapy, and nucleic acid therapeutics. Then we have to discuss technique used in pharmaceutical biotechnology, that is PCR, DNA sequencing, affinity protein purification. Then fundamentals of genetic engineering and its application in medicine, we have discussed a lot about it. Then pharmaceutical recombinant DNA technology, therapeutic proteins, growth factors and therapeutic antibodies, high throughput screening of putative therapeutic compounds. Now, biotechnological aspect in product development. We did discuss a lot about it, and we discussed various 6 phases through which we would be able to get most modern medicine, what we call it as molecular medicine. Now, principle, synthesis and application of monoclonal antibodies. We did discuss a lot about it while we were sharing about it. That is, they are the most important aspect of the molecular medicine, which is being used particularly in cancer therapy, and it's very costly, and mostly the Jews are being used it in a lot, and they are acquiring, and the day will come it will be available in Pakistan as well. Then the immobilized enzyme and their application in medicine. This is the dilemma of the talk of today. Immobilized enzymes and their application in medicine. Now you see, what are the various kinds of enzymes which are used in medicine? Can you see TPA, that is tissue plasminogen activator, streptokinase, so and so forth. These are the enzymes which are used in cardiovascular disease. TPA is the most modern one, tissue plasminogen activator. This is being used in myocardial infarction. Whenever the heart is infarct, and if you could see, this is the heart, and heart has been infarct herein, and plasminogen is not going to be released. Now, if you could administer into it TPA, immediately heart will cure and patient would survive, and within 2 hours patient would go to his home very quickly. He would be very okay. So this is the most modern way which could be able to cure the diseases, particularly heart diseases and so forth. So in that respect, I would like you to know herein immobilized enzyme and their application in medicine. Now, I would like to explain it. What do you mean by immobilization? As the name indicates, immobilization. Im here means now, mobilization to mobilize. So it should not be mobilized. It should be restricted on a particular area, and in that respect we could know it. That is, if here is enzyme, it acts upon the substrate and we will be able to get the product which is in the form of enzyme or any other protein and so forth. So looking into it, the important issue herein is enzyme is there, it acts upon the substrate. The word is used immobilized. Enzyme should remain immobilized herein on the substrate, and the substrate has to be converted into the product. So the important issue is the substrate should be of such kind that enzyme could be able to stay upon it and product is formed. and how we could do that the enzyme should be immobilized here and it should perform its function and it could be able to give the product which is required. So there are most modern techniques which are being used under the aspect of pharmaceutical biotechnology. Those are the, depending upon the chemical structure of the molecule of the substrate, that is as follows. Introduction, immobilization, surface immobilization by covalent coupling. Herein you can see, if this is the substrate, it has got its surface and the enzyme should be able to make covalent bonding herein, so that enzyme should be restricted upon the substrate and we would be able to get the product. So the covalent linkage would be able, a pretty good technique wherein the enzyme would be restrained, enzyme should be made immobilized on it, so that pretty good product will be manufactured and in the form of medicine, and that will be used and we could be able to cure the heart diseases and multiple other diseases which could be done in this way. So this is one of the ways, that is the substrate is therein. Now under this you can see various kinds of polymers and other compounds are being used. In it we can use polymers with hydroxy group and we can use base protein coupling, covalent coupling employing in N-succinamide activation, coupling through diacyl fatty anhydrates, and shifts base reaction and periodate activation, and carboamide mediated coupling, and conjugation through phenolic group of protein or enzymes, conjugation through sulfhydryl group, application of covalent and coaching, and last and least in avidine biotin affinity group. Now the second aspect of it is, that is, what could be the substrate? Substrate could be adsorption. Phenomena should be adsorption herein. So if this is the enzyme, enzyme should adsorb upon the substrate, adsorption. It is the way that it should remain upon it. And if you could see, how does we could use the charcoal in the medicine? Now if this is the charcoal particle and toxic substance are therein, and the toxic substance will adsorb upon the charcoal and it will be removed. Now similarly we can use herein the enzyme should be remain adsorbed and restricted upon the substrate, and as it will be retained herein and restricted on this area, it will be able to acting upon the substrate and would be able to produce a product which is the desired response, and we could be able to get the pretty good protein or pretty good enzyme which is required and we can use in the medicine. For example, you can say various other enzymes which are biologically important. Now under the adsorption we can see forces involved in internal surface immobilization. Methods for immobilization by adsorption. Immobilization on self-assembling nanocrystalline ceramic core. Then this was about the adsorption. Now the next task is the complexation and chelation. Now this is important in a way that we have to restrict the enzyme on the substrate. How we would be able to do? The next task in this response which we are going to do is, that is the complexation. This enzyme would form a very complex structure with the substrate. Depending upon the chemical structure of enzyme and upon the chemical structure of the substrate, it will make a chelate with it and let the enzyme to be stay upon the substrate and to get the product. So under this there is metal chelation. You can use various kinds of metal which make the chelate with the enzyme. Then chlorocomplexation of titanium in HCl solution. Then hydrox transition metal oxidase. So this was the way through which we can make various kinds of chelate. Now the next, in this respect, last and least, is with sport immobilization. That is, enzyme is there, act upon the substrate, some sport should be there and on which the substrate should stay and enzyme should act. In here you can see the chemical reaction is not involved, but it is just a sport which is required, and the substrate will not mobilize and enzyme will not mobilize and it will remain on the substrate. This is then we can entrap the enzyme on the substrate. Just entrap upon it, no chemical reaction, no biological reaction, no any kind of reaction is being involved in it. It is just physical entrapment wherein the enzyme will stay upon the substrate. Now the microencapsulation, that's another way, that is we can use microencapsulation as we have been using in the medicine, the most modern way to get medicine. So we will be discussing these one by one. The cell mobilization is not our area. Our area is to restrict upon this aspect, that is immobilization, surface immobilization, covalent coupling, by adsorption, by complexation, by chelation, and within sport immobilization. Up till here we should have to discuss. Now we are going to deal with, first of all, surface immobilization by covalent coupling. Herein the enzyme has got a chemical structure in a way that it has to form a covalent coupling with the substrate, so enzyme should stay there and ultimately the product will form and we will be able to get whatsoever is required in the form of medicine. Now in here, previously I did discuss the first method which is used to sustain enzyme upon substrate is surface immobilization by covalent coupling. The covalent bond between biocatalyst and sport matrix form a stable complex with the appreciable stability during functional use of enzyme. However, the process subjects the delicate three-dimensional structure of the biopolymer to strong disruptive physical and chemical forces or stresses. Therefore, where superior form and stability of immobilized enzyme overweighs the irreversible loss of activity that inevitably associated with immobilization process, it is referred over soluble form. Now, how does that can be? The functional group present on enzyme protein through which a covalent bond with the polymeric or other supports could be established should be non-essential for enzymatic activity. That means the enzyme should not be altered. It should be of such kind that the enzymes should remain the same as it is. Its nature and its kind and its physical and chemical structure should not be changed. Though moreover, the reaction which involved relatively mild condition and essentially utilize and aqueous media are preferred. Here is that is the aqueous media should be there so that there should be formation of covalent linkage established in a way that the enzyme should be stay upon the substrate as it is referred. This further necessitate the complete knowledge of amino acid and their active group. You can use subhydral group and the fifth one in that case is phenolic component could also be used in that respect. Under the NH2, if you could see in here, the lysine is the most important N-terminal of polypeptide chain could be used. Beta and alpha of aspartic acid. It is an acidic amino acid which contain two acidic group in, and the glutamic acid respectively. Again, the glutamic acid is an acidic protein. It also contains two acidic group in it. Then phenolic ring on tyrosine. If you could see the tyrosine, the structure of the tyrosine contain phenyl ring, along with that one OH group is attached upon it, and sulfhydryl group of cysteine. If you could see the cysteine molecule, this contains sulfhydryl group in it. Now group on serine, tyrosine and threonine, which contain this group. This is important in this aspect. Furthermore, on the amino acid, the groups which are present, that is indole and imidazole, and the structure of is as follows. So the indole and imidazole, here is the structure of indole group and this is imidazole group. These are also important, which are present in amino acid and responsible to let the enzyme to be stay upon the substrate and having a covalent linkage. On the other hand, the polymeric sport, which have been explored as sport for covalent immobilization, include hydroxyl group of polysaccharides, amino acid and related groups of amino ethyl coated polysaccharides, then carboxylic acid and related group of polyglutamic acid, maleic acid, and so forth, aldehyde and acetyl group of polymers, amide group of polyamides and polyamides. And now the next we are dealing with the polymers with hydroxyl group. We will be discussing these later on. Thank you very much. If any question is there, you can read it out and let me know. Actually, I am again going to repeat it, that we are sharing with how does we will be able to produce biological enzyme. And you see the enzyme, how does that produce? Enzyme act upon the substrate and product is formed. And we can let the enzyme to stay upon the substrate and act upon the substrate so that the product will be formed. For that matter, we would let the enzyme to stay by different ways, by covalent linkage, by adsorption method, by chelate formation, and by complexation, or sometime we would have to have a physical sport upon it so that enzyme should stay and produce the product, and that product, which is in the form of enzyme, could be used in the medicine such as TPA, tissue plasminogen activator, and so forth. My dear students, if you feel like any problem, I am in front of you and you can ask me on my mobile as well. You all have got my mobile number, and that is, I can repeat it for you, 0300416375. You can talk to me anywhere, whenever you like. Thank you very much.